Hello, everyone, and welcome to our one-on-one discussions with the scientist. Today, it's my pleasure to speak with Dr. Helen Bramlett. Dr. Bramlett is professor in the Department of Neurological Surgery and a very important member of the Miami Project to Cure Paralysis. My name is Dalton Dietrich, and I'm the scientific director of the Miami Project. So, Helen, welcome to the program today. How are you doing?
I'm doing fantastic, Dalton. Thank you for asking.
Okay, very good. So Helen, you're a major researcher in the Miami Project. It's interesting that, of course, in the Miami Project, we focus on different types of neurological disorders, and you work on spinal cord injury, traumatic brain injury, concussion, and stroke. Now, how do you do that in terms of the scientific questions that are important in each of those different, apparently different disciplines?
That's a really great question. I have a fantastic staff and excellent collaborators on all three of those areas of CNS injury. And through them, we're able to basically organize ourselves, and we work on different aspects of each area, whether it's brain trauma, stroke, or spinal cord injury. And I could not do this research without them and my colleagues at the Miami Project as well.
So most of your research is directed toward looking at neuroprotection. Is that correct? And trying to understand pathomechanisms that can be targeted for therapeutic interventions?
Right, that is correct. Most of the studies, whether they are in stroke or spinal cord injury or traumatic brain injury, we're looking at not only just pathophysiology of those different types of CNS injury, but we also look at a lot of different therapeutic interventions. As you know, we've been doing a lot of work over the past several decades in the use of therapeutic hypothermia and targeted temperature management. And obviously in the late 80s, we were the first group to actually show an improvement in a preclinical animal model in terms of cardiac arrest after cardiac arrest. So, and this led to eventual changes in guidelines. It was translated to the clinic. And so our goal now is to translate anything, whether it be therapeutic hypothermia or some other type of drug, have it translated into the clinic.
And that's been shown in traumatic brain injury, stroke, and spinal cord injury. And your laboratory and colleagues have actually contributed to a lot of that preliminary study that's led to these subsequent clinical investigations.
Exactly. In fact, we had a paper that was published several years ago showing that if you use therapeutic hypothermia initially, you could reduce seizure activity long term and after traumatic brain injury. As some of you know, the incident rate of seizures or post-traumatic epilepsy increases for those individuals who have had some type of early brain trauma. So these seizures, they'll pop up maybe 10, 15 years later after the initial insult.
Mm-hmm. So targeted temperature management is an extremely important clinical question that's being pursued. Some of your work with your colleagues have also looked at recently concussion and the importance of temperature in terms of looking at the pathophysiology and outcome in concussion. Can you tell us a little bit about the importance maybe of fever or hyperthermia?
Right. So we've done some of our more recent work has looked at hyperthermia or elevated temperature. You only have to raise temperature a couple of degrees. 37 Celsius is normothermic. You only have to raise it to about 39 degrees Celsius. And it's easy for someone who is out in a hot environment excuse me, and in terms of raising their body temperature up to 39. And so what we've done in the lab, we've tried to mimic that and our concussion model. So it's not, you don't produce gross pathology in the brain, but you produce a type of concussion type insult. And then we actually elevate temperature prior to the concussion, because many times you have soldiers who are out in the field and they've had undergo some type of blast trauma, for example, as well as athletes. They're out either in practice or they're actually in the game and they have some type of concussion. And most likely their body temperature is elevated to a hyperthermic level.
Yeah.
So
You
yeah,
built-
so we're looking at the pathophysiology in terms of that. And we're also looking at possibly, so someone comes in, they're hypothermic, they've had a concussion, reducing temperature to either normothermic levels or actually reducing it further to therapeutic hypothermia for it to act as a neuroprotective mechanism.
Very interesting. You mentioned soldiers, and it's important to note that you're a research health scientist at the Department of Veterans Affairs, associated with the Miami VA, right across the street from the University of Miami. How do you deal with that, and how does that relate to some of the military questions that are being asked with soldiers?
Right. So I've been at the Miami VA for over 10 years now, and it's a very important administrative unit of the federal government. They, not only do they take care of the veterans that have left military service, but they also provide an avenue for not only clinical research but also for basic science research, and which is what I'm involved with. I have again excellent collaborators at the Bronx VA. Two of the members of the Bronx VA, our studies are involved in spinal cord injury. So these are my two spinal cord injury grants through the VA. And we are looking at not only at whole body vibration, which is a possible something that could be done at your home to improve function after spinal cord injury. There have been several studies that have been published clinically, and so we're looking at more of the mechanisms and stuff, and what is present or what's activated with whole body vibration in terms of improving functional outcome in combination with other drugs. And then the other study will be also assessing muscle activity, muscle atrophy after spinal cord injury as well, using a therapeutic drug.
And that's so important in the spinal cord population, because we lose muscle mass very quickly after spinal cord injury, and there's also skeletal muscle. So the whole body vibration is actually a way to maybe even enhance recovery and protect these other organs after spinal cord
Exactly.
injury.
We've also seen, reductions in bone loss that also happens after spinal cord injury, obviously because these individuals are in wheelchairs and they're not moving obviously as much. And so these preclinical studies are very important in order to translate our work into the clinic, which is, I think, one of the missions of the Miami project. And we have been able to do that, with therapeutic hypothermia. There's a Dr. Alan Levy, who's the chair of neurosurgery, is a PI on a DoD grant that's a multi-center clinical trial looking at the use of therapeutic hypothermia after spinal cord injury.
Mm-hmm. Speaking of spinal cord injury, one of your other programs has to do with BrainQ. It's a company in Israel, and they're working on these new neuromodulation approaches for spinal cord injury, and you're involved with the preclinical studies. Can you kind of tell us about BrainQ and what that concept is?
So the basic concept of BrainQ is they took a lot of different electrophysiological recordings, okay, let's say from subjects, and they modified their device so that they would be able to activate certain circuits in the brain using different frequencies. And depending on how long you activate it, hopefully you will be able to show recovery after some type of injury or disease. And they had done some preclinical work and a little bit of clinical work in this, and they had approached us to take it a step further in terms of our more clinically relevant contusional model of spinal cord injury. So that's what we're doing on the preclinical front. We are, after spinal cord injury, these animals are placed in a device, and their electromagnetic, different electromagnetic frequencies are sent into their bodies basically, and then we test them for behavior. And what's really interesting is that in concert with the preclinical work, downstairs they're actually doing the clinical work as well, and a clinical study with a couple of other institutions in the United States, and So we've got two preclinical study and a clinical study going on with BrainQ. It's really exciting work. We're really happy that the company approached us, and we think that, you know, we've seen some preliminary data and it's looking pretty good, and so we are continuing on with BrainQ.
Fantastic. Yeah, we've already heard from Dr. Katie Gant about some of the clinical studies in BrainQ, and they're now doing that remotely because of the COVID-19 pandemic. We're having to actually do these studies at their home, which is very exciting because this is a technology that we can actually take home. So, okay, so your research is extremely exciting. In addition, you are director of the undergraduate neuroscience program at the University of Miami. So tell me about what those responsibilities are. And during this period of time of this pandemic and people physical distancing, how can you actually teach students actually the neuroanatomy that you teach? How can you do that in a distant situation?
So it's been very interesting. So we have been teaching remotely about a month now across the university, both for our undergraduates and our graduate students as well. And the students that I teach a neuroscience lab course, and my portion is neuroanatomy. So you might ask yourself, well, how are you going to teach neuroanatomy